Hello everybody, my name is Dr. Sarah Guerin from the University of Limerick, and thank you for coming to today's webinar, which is on the modelling of anisotropic phenomena in pharmaceutical crystals. So a little bit about me. I'm going to bring up my laser pointer. I am a second year postdoc in UL in SSPC, me over here. My undergraduate degree was in applied physics, but my PhD and my first postdoc were in the predictive design of biomolecular crystals. And that just means that I was using the same models I'm going to be talking to you about today, but for electronics applications. So it was amino acid and peptide crystals, so small biomolecules. And not only was I using my molecular models, I was also growing and characterizing these crystals in the lab. So what I was doing was developing highly quantitative models, and I'll show you some more information on that in a while. So directly predicting the numbers that we wanted to measure in the lab as accurately as we could. And then constantly creating a feedback loop of experimental verification, both in UL and collaboratively with other groups around the world. And what these electronics applications were for these biomolecular crystal energy harvesters, and we could predict things like how much voltage, how much energy you could get out of a biomolecular crystal device, and things like their mechanical properties, which I'll be talking to you about as regards pharmaceuticals. So the three key messages I want you to take away from today generally is about molecular modelling. Firstly, that we're using a nanoscale insight, the smallest possible lens, to look at atoms, molecules, electrons, to firstly understand, but then also engineer and control macroscopic properties and industrial scale processes. So predicting the behavior of a material, but also predicting how that material will interact with its environment, say in a manufacturing setting. Secondly, as I've said, highly quantitative. We don't want to just tell you that something is, that say one crystal is better than another crystal. We want to be able to say, no, this crystal is going to have a Young's modulus of 10 and this crystal is going to have a Young's modulus of 2. Exactly, like I said, what you're going to be measuring, either in a lab or in an industry setting. And then finally, that molecular modelling has broad applications and ever-growing potential. I'm talking to you about such a small subset of what these models can do today, and really it's about creating a dialogue again between us, experimentalists, other modelling groups and industry, to expand the applications of these models and constantly build, improve and strengthen what we can do. And if you want to hear much more about molecular modelling as a whole, and how it can be used to design the next generation of pharmaceutical crystals, you can watch the previous webinar on the SSPC website by my supervisor, Dr. Damien Thompson, if you want to check that out. But what I'm going to be talking to you about today is the specific models that I've been developing since way back in 2016 to predict the properties of single crystals using something called density functional theory, which is, very simply put, just using quantum mechanics to predict the properties of materials. So everything that I've done up until now has been on single crystals, and just what this graph is showing you is just the high level of accuracy between the numbers that we predict and the numbers that we see in the lab. And this spans inorganic crystals, biomolecular crystals, both amino acids and peptides, biomineral crystallizations, things like calcite, and then just as we started into SSPC here, we were starting to look at cocrystals and coformers. So that's the level of accuracy that we want to achieve in our predictions, and something that we're continuing to bring to our pharmaceutical crystals. So how can we integrate these quantum mechanical models into what SSPC does? So what we've developed over the past year is three key areas where we feel that we can do this, and these are the three sections of the webinar today. So firstly, we're going to look at investigating molecular interactions in manufacturing processes, so how we can study how all the different molecules in any kind of batch process where you have lots of different ingredients, and we'll talk about the specifics in a while, how we can study very simple interactions in order to determine what happens in those large-scale processes. Following on from that, I'm going to talk to you about tracking properties through co-crystallization, so studying the properties of just an API crystal and then seeing how those properties change when they're crystallized with other molecules and how that affects their properties. And then finally, I'm going to talk to you about the white whale of modelling, predicting crystal solubility, and how if we study this as an anisotropic phenomena, we might be able to open a new window or provide some new insights into these in vivo phenomena. So, as modelers, we can't exist in a vacuum, we can't stay on our computers all day. There needs to be a constant feedback loop, a constant dialogue, or as you can see here, a workflow between what we do in terms of predicting and rationalizing behaviors at the nanoscale and what happens in the lab or in an industrial environment. So when we were setting up this workflow, we were at the point where experimentalists had already grown and characterized their crystals, and they wanted to know the atomic underpinnings of what they'd already seen. And this allowed us to build up a large enough data set that we can now begin to start, as we like to do for predictive design, with predicting the properties first in order to guide experimentalists along their own workflow. So as I'll give you some case study examples, if we take a set of 10 potential co-crystal or co-former API combinations, that hopefully we could provide some insight into what the inverted commas best crystals from that data set would be in order to focus time and resources, so that we'd start with our predictions and then go through the workflow of the experiment and the characterization, hopefully moving off into moving those crystals further along the line, while also rationalizing the behaviors that we're seeing in order to go back to engineering crystal properties. So if we know enough of what happens in the unit cell of a crystal that gives it, say, again, a desirable Young's modulus, or good bioavailability, or good solubility, then we can look at actually designing unit cells to give specific properties and then moving on to grow those in the lab. So I put a nice quote here that I like, just highlighting that we need to combine theoretical, computational, and experimental work in order to develop predictive tools for crystal growth and dissolution, which is one of the biggest goals right now in modelling of pharmaceuticals. So the first section that I'm going to talk on today is investigating molecular interactions in manufacturing processes. And in order to do this, I bring you into the world of excipients. So generally when we talk about pharmaceutical crystals, we're talking about our active pharmaceutical ingredients, our APIs, and if we're looking at co-crystals, we'll also have our co-former molecule. But what we also have, if we're actually making a drug or making a tablet, is the excipients. So firstly, just in case you don't know, the role of excipients is generally to aid in the processing of the drug during its manufacture. And in some cases, it can actually protect, support, or enhance certain properties of the drug. More often, it's used to assist in the delivery of the drug, or to maintain the integrity of the drug during storage. And in order to facilitate these roles, there are a number of key properties that excipients need to have. They need to be very stable, which is something that we can look at with our models, and very reproducible, minimal interaction with the drug, which I'm going to be talking to you about in a second. So we don't want our excipient molecules and our drug molecules to be interacting. We want them to be pharmacologically inert, so they're not doing anything once they get inside of the body, and they have their desired functionality and that they're cost effective. So where do we come into this as molecular modelers? So our aim, we want to study simple molecular interactions in order to rationalize large-scale manufacturing processes. So what does this mean? Let's look at a real-life example. So in this case study, we were looking at the drug THP, which is used in therapy for respiratory diseases such as COPD and asthma. And this was being co-crystallized in the presence of 4-ABA, which is 4-amino-benzoic acid. And again, that's a common co-former molecule that's been approved for co-crystallization with other drugs, such as antibacterial drugs. So the co-former and API combination were milled in the presence of 5 tablet excipients. So these are our 5 excipients here. Some of them are semi-crystalline, some of them are amorphous. So we have multicrystalline cellulose, lactose, PVP, HPMC, and PEG. And again, what was interesting, and you'll see in the next slide, is that we were contacted after the initial experiments had taken place that were showing that co-crystals were being very successfully processed with HPMC and lactose. And again, we wanted to see if there was a molecular underpinning for that and if we could rationalize what they were seeing in the lab. So, also a great case study on not expecting the model to confirm what you're seeing in the lab. So this was the initial order that was brought to us, so that we hoped we'd be able to rationalize HPMC and lactose on top. But when we ran our binding energy calculations, which was looking at this is our API and co-former molecule, and this is our excipient molecule, and this is how they would see each other in that process. It's all powder based, so you have your free molecules interacting with each other in this manner. And when we ran these binding energy calculations, we saw that PEG theoretically should be giving the most stable co-crystals, and then HPMC, lactose, PVP, and multicrystalline cellulose. And when we reported this, the experiments were run again to reveal that there was just a processing error with the PEG, and that it did indeed form the most stable co-crystals. So what were those calculations actually telling us? So we were looking at the binding energies. We were trying to see if any strong interactions occurred between the API and co-former molecule, looking at various different configurations to see if they would bind strongly to the excipient molecules. Because if the excipient binds to the co-former and the API at the start of processing, then it won't be able to interact with the other API co-former complexes to form a co-crystal. So you have all these molecules, but if they're all sticking together at the very, very start of the process because they have very high binding energies, then you're not going to get your nice co-crystals at the end. So there was a number of different inter- and intramolecular interactions that we could look at, which contributed to our binding energies and could tell us which excipients would produce the most stable co-crystals, which really was about the excipient interacting in the smallest way possible with the API and the co-former. And that's what I was saying in 1 or 2 slides ago when I was saying they have to be inert and they have to not react with the API and the co-former, and that's what we can study. So these would be some of the nicest and the simplest models that we can build, just looking at 3 molecules here interacting with each other, and it can tell us so much about what could be a very large scale manufacturing process between APIs, co-formers, and excipients. So the next thing I'm going to highlight is I'm going to go straight into crystals and talk about tracking the macroscopic properties through co-crystallization. And again, what does that mean? Let's look at some examples. So why do we need co-crystals in the first place? One of the main reasons why certain drugs don't get through all the way to actually becoming pharmaceutical products on the shelf is poor solubility and low bioavailability. But what if we could take those drugs before they hit the trial process, and what if we could improve those properties by introducing another molecule to crystallize with your API? And it's been found in most cases that when you co-crystallize a drug molecule, you can develop superior macroscopic properties. And some of the key ones I'm going to be talking about today are tabletability, solubility, and stability. So again, we can predict the properties before co-crystallization and after co-crystallization. So simply put, this is our equation for making a co-crystal. Our active pharmaceutical ingredient are paracetamol or our ibuprofen or our THB, a co-former molecule to make our co-crystal, and we want to improve the material properties while preserving the pharmaceutical properties. So we still want paracetamol to be paracetamol, but maybe we want it to react faster in the body, react slower in the body, take on a different form, and we can do that via co-crystallization. So I'm going to talk about 3 co-crystal studies that we're currently or have been working on. And firstly, I'm going to talk about the case where we have a number of pharmaceutical ingredients and we're testing them all being co-crystallized with the same co-former. So this was a set of crystals that were brought to us by an SSPC member. We had studied amino acid crystals in the past without any co-former, and this postdoc had grown all of these crystals with sulfamic acid as a co-former. And what we wanted to do was predict which crystals have the best properties, and as I mentioned at the start, what crystals should that postdoc then focus on? These were particularly difficult crystals to grow, so obviously it's better to focus your time and your resources. If it's difficult to grow 12 crystals, then it might be better to focus your energy on growing 3 very difficult crystals. And again, building up our knowledge on what fundamental nanoscale properties of crystals make a good crystal as regards what an experimentalist or an industry researcher is looking for. So what we did in this case is we looked at all the different properties and we narrowed it down to 3 candidates where co-crystallization improved the physical properties, but for the other 1, 2, 3, 4, 5, co-crystallization actually deteriorated their properties. So that way the time and the resources could be focused on the co-crystals where the physiochemical properties were improved, and then characterizing and studying those crystals further. In terms of the number of atoms, just a key point to mention for our particular models, there is a limitation on the size of the systems that we can study. So this is why I would have a table like this. And say for the binding energy calculations that I showed you with the excipients, we could go quite large with our unit cell. We could go maybe 600-700 atoms with the kind of modelling that we use. But if we're predicting highly anisotropic properties, things like elastic stiffness, dielectric behaviour and things like that, then we're kind of capped at the lower end of this table here. So 260 would be quite doable with the resources that we have on the national supercomputer, but 385 would be quite a challenge just in terms of computational time, computational power. So the second case study that I want to show is demonstrating exactly what the title of this section was, which is tracking properties from API crystal and coformer crystal to the final product. So in this case, our active pharmaceutical ingredient was N-acetyl-L-alanine. So it's just an alanine derivative. This is its single crystal. This is one molecule here, and you can see the blue lines just showing the hydrogen bonds between them. And what was unique about this is that we had the crystallized API, but we also had the crystallized coformer. So this is a BPY coformer molecule. Again, common simple molecule that's used as a coformer. And these are just waters of crystallization. So there are structural waters in the crystal. And then we had the Cocrystal. This is just looking at it from two different views. It forms this really unique pore structure. And what we could do is we could look at the induced polarization, we could look at the lattice energies to see how stable the co-crystal was, and we could also look at their mechanical properties. So we could see what does the coformer like to do by itself, what does the API like to do by itself, and what happens when you put them together. And by tracking those properties through, again, we can start to look at engineering co-crystals and different API coformer combinations. So in this case, we can look at things like polarization, which is connected to solubility. We can see if the co-crystal is stable. Will it be something that can actually be grown in the lab? If we had a variety of coformers or APIs, we could rank them in terms of stability, and also screen coformers in that way. And even simpler than that, we can have the same active pharmaceutical ingredient, which is our alanine derivative. We can have a coformer, BPE, again a very simple molecule similar to BPY, again forming these really intricate porous co-crystals. But in this study, all we did was we changed the chirality of our active pharmaceutical ingredient. So we had L-alanine, D-alanine, and then the racemic mixture of the two, so DL-acetyl-L-alanine, or DL-acetyl-N-alanine. There we go. So what we learned from this was simply by altering the chirality of the molecule, so whether it's a left-handed or right-handed version of the exact same molecule, mirror images, we actually see a massive change in the macroscopic properties. So the L and the D active pharmaceutical ingredients, they form more flexible co-crystals, which is desirable for some applications, but in terms of tabletability and processing is less desirable. But when we have a mixture of the two, when we have D-alanine and L-alanine, we get co-crystals with very high mechanical strengths, so Young's modulus of 50 gigapascals, which would be closing in on kind of metallic level of mechanical stability. So, as I said, the mechanical properties are key in processing, things like powder compaction. And what we do here is we don't just extract a single Young's modulus, we actually get an 18-component elastic stiffness tensor. So we can examine the directionality of the Young's modulus, the anisotropy, how it changes in different directions. So the mechanical strengths along one crystallographic axis can be very different to the strengths along another axis, and that can again inform our manufacturing process. And we can also look at things like shear planes, what happens if you shear the crystal versus just applying a standard longitudinal compression. And again, that builds up our knowledge for a number of ways to engineer the properties of co-crystals. So the models that I've been talking to you about today, using density functional theory, quantum mechanical based models, we can't directly model solubility with this because we can't study temperature effects. So we can only look at crystals pretty much at 0 Kelvin. And to get our full solubility curves, we need to study phenomena over a range of temperatures. So the models that we can use for that are actually being carried out by my colleague Pierre Cazad, who's also an SSPC postdoctoral researcher, and that's using molecular dynamics models, which can study crystals in large amounts of solution over a range of temperatures. So in that way, we could be able to predict directly the quantitative solubility of a crystal. But in terms of what my models can do, what DFT can do, we can try and extrapolate from what we know about molecular solubility what makes a crystal more or less soluble. So we know that in terms of molecules, like amorphous materials, the solubility is dependent on electrical properties like polarizability, dipole moment, HOMO-LUMO gaps. And what we'd like to see is if you can extrapolate these kinds of behaviours, but in terms of crystals. Now, this isn't saying that a soluble molecule makes a soluble crystal, because we know that's very much not true. You can have a highly soluble molecule, but when it crystallizes together, all of those intermolecular interactions can completely change the macroscopic properties. We've seen that in absolutely everything that we've studied. So that's not what we want to do. What we want to do is take something like, say, the dipole moment of a crystal, which will be essentially a vector sum of the dipole moments of the individual molecules, depending on the supramolecular packing of the crystal. So in that sense, can a net crystal dipole moment correspond to a smaller or larger solubility? And we've two interesting case studies of this. We have quercetin and EGCG, both widely studied drugs, both studied or widely studied by SSPC researchers, and there's about an order of magnitude difference in their solubility. So EGCG being the molecule in green tea, very low solubility, and quercetin, highly soluble crystal. So we can study the electrical properties of these crystals and see if we can start to see clear trends in the inherent unit cell properties that contribute to large or less large solubility behavior. And there are two ways we can complement this kind of molecular underpinning or rationale for what makes a soluble crystal. The first can be in situ observations of crystal dissolution. So some great studies coming out of UL right now from our Titan TEM studying the growth of pharmaceutical crystals at the smallest possible scale. So it would provide a lot of insight, again, in terms of how directional is solubility. Where does dissolution begin? And again, not only predicting these net crystal properties, but again, studying them using our tensor methodology. So things like polarizability can have up to 18 components for a single crystal, because you can generate things in longitudinal directions, but also along shear planes. So how directional is this response? And do you only need one, say, highly polarizable crystallographic axis in order to have high solubility? And understanding mechanisms like that would give us a lot of insight into engineering more soluble crystals without even resorting to co-crystallization. But also we'll have our direct solubility output, and that will be from Dr. Cazad in terms of our thermodynamic cycles. So we'll have our direct solubility prediction, but then we'll also have the why behind that. And again, I've just taken this nice quote here. The literature is pointing to this idea of directionality in pharmaceutical crystals, that it's important for understanding these complex phenomena, that we should be incorporating it into our molecules or into our models, and how we study pharmaceuticals, and also how we process pharmaceuticals. So not just looking at Young's modulus, but looking at an elastic tensor, and not just looking at dielectric constant, but looking at a dielectric tensor or a permittivity tensor. And by using that directionality, it can guide us in the right direction as well. So that's all I want to talk to you about today. I'd just like to thank all of my research group and collaborators in UL, and then my further afield collaborators, as well as iCheck, our national supercomputer where we run absolutely all of our calculations. We wouldn't be able to do any work without them. And I'd like to thank you for... you can contact me or find me absolutely anywhere. I've left some of my details on this slide, and I'd be delighted to answer absolutely any of your questions. I hope I've given you something to think about, given you some insight into molecular modelling, and I hope that you learned something. Thank you.